So I'd like to first thank all the kind people that have helped me through the years. People who worked in my lab, and a good number of them are in the audience, which is really nice that people have. But I wanted to start out with that before I got too carried away. I hope some of you found my title somewhat ambiguous and obscure, and so there's two ways to interpret challenging science. One is, does Bloom find science challenging? And yes, indeed I do. Or do I challenge science? And indeed I do. And so I want to give you some examples, since this is supposed to go back to the history of my scientific career there at Davis. And so I'll be covering several topics. One is the model control. The other is CAM and C3 carbon fixation, nitrate versus ammonium as a nitrogen source, and carbon-nitrogen relationships. I want to start out with a review we wrote, opinion piece we wrote for a journal that was now extinct, but it's what's new in plant physiology. I guess it's extinct because there's nothing new in plant physiology anymore. But this was something I wrote with Eduardo Zeiger and Peter Kepler. And if you read it today, you say, why they ever publish it? It's just something, what we were proposing at the time is that potassium influx into guard cells that causes their opening was driven by a proton gradient, by pumping protons out of the guard cells and using the osmotic gradient to drive in potassium. Now, today that would seem just trivial, but at the time it was inflammatory. In fact, we had the leader in the field wrote a review when we submitted it. His name was Klaus Raschke. He was at the Michigan State University DOE lab, and he wrote, what we needed are more data and fewer discussions made over beer. About a year after this, he left Michigan State and took a professorship in Germany, and then spent the rest of his career making discussions over a beer. So, the second thing I'd like to show you is my thesis work that I wrote, CAM versus C3 carbon fixation, at a paper in Plant Phys. I was intrigued by the fact that crassulation acid metabolism was found in a wide range of taxonomic classifications. It was spread over the whole plant kingdom in a way that didn't make sense. There are at least 20 different independent origins. And this was similar to C4 carbon fixation, which is now considered to have arisen 60 times independently. So what I did was I emphasized, which you may be not able to read, but the fact that CAM and C4 are exaggerations of the malate shuttle that occurs in all plant species and all plant tissues. And this was published in Plant Phys in 1979, which seemed quite reasonable, but it again inflamed the crowd. And so about 3 years later, after I left the field, this group wrote a Evidence Against Bloom's Hypothesis, was the title, which I don't think Plant Phys ever does anymore. You see I'm cited right there in the abstract. And what they did was put up a straw man. They said I had stated things that I never stated. And then they attacked the things I had never stated. And I wrote the editor saying, can you ever read the original paper that they're attacking? And they said, oh, you can't, we won't publish any letters. We don't publish letters in physiology. And I've moved on. So we have the thing that I started when I came to Davis was looking at ammonium and nitrate as nitrogen sources. And this came out of my feeling that 95% of plant papers that deal with nitrogen treat it as a single entity, as a monolith, and it's called total nitrogen. And they don't even mention that nitrogen comes in different forms, and the nitrogen forms differ so distinctly from one another, they should be considered separate elements. So I began looking at the forms that are most available to plants, and that is ammonium and nitrate, which are sort of the yin and yang plant nutrients. And if one compares them, one gets an idea of the extent of the difference between these various forms. Ammonium in high levels, if it's not assimilated, and we'll talk more about this in a second, is toxic in tissues because it decouples pH gradients. Nitrate is benign in tissues, can be stored in very high concentrations, and can serve as a counterion for potassium and other cations. In fact, it's a metabolically friendly osmotic for those of you who are interested in water relations. Ammonium is a cation, binds to the cation exchange complex of the soil very strongly, where nitrate's an anion that's more mobile in the soil. Most soils it moves very freely, and hence the problems of nitrate leaching. Ammonium is, because it's binding to the soil cation complex, it's usually found in very low concentrations, freely moving. Nitrate is in relatively high concentrations. Ammonium seems to be the most important nitrogen form late in forest succession, where nitrate is more important earlier in succession. Most microorganisms strongly prefer ammonium as a nitrogen source, where they will use nitrate, but only under duress. In fact, many microbacteria will start to sporulate if you force them to use nitrate as a nitrogen source. And the reason for this is that ammonium is a much higher form of nitrogen than nitrate. In fact, its oxidation state is +3, where nitrate's oxidation state is -5. An enormous difference in the energy of these two forms, and we'll go into that in a moment. So why is ammonium toxic? And I make this like a kid in a candy store, that if you give a plant too much ammonium, they'll eat too much, they'll absorb too much, that they can't assimilate it, and they'll get sick to their stomachs. More detail and a little bit more scientific, here is what happens to ammonium in tissues if it accumulates to any degree. That in systems where there's a pH gradient that's developed, and there are some such here, I give the examples of the stroma of the chloroplast versus the lumen of the chloroplast, the matrix of the mitochondria versus the inner membrane space, or in a cell, the cytoplasm versus the vacuole. These are all compartments separated by a biological membrane in which a proton gradient is used as a way of storing energy. And that the presence of ammonium and ammonia, the interconversions of ammonium into ammonia and ammonia's membrane permeable will actually dissipate that proton gradient and therefore dissipate energy processes in cells. So we began to look at what happens to plants when they're actually given non-toxic levels of ammonium. Most studies of plant nutrition have made the mistake of giving ammonium at toxic levels. So those of you who use MS medium or full-strength Hoaglands may realize that you're giving them 21 millimolar ammonium. It was a medium that were developed for tissue culture, not for plant nutrition. And 21 millimolar is about 1,000 times higher than they need for full growth. It allows you to be lazy, you don't have to punish your nutritional medium very much, but it does subject plants to toxic levels. So we started looking at what happens to plants when you give them non-toxic levels, and here is tomato plants grown under ammonium versus nitrate. What you can see in the first column is that the growth of the two biomass is equivalent under either form, under non-toxic levels. Plants under ammonium tends to develop a larger root system, and this makes sense for a less mobile nutrient, such as ammonium, which binds to cation exchange, versus nitrate that's fully mobile. You don't need as large a root system. So that's one thing we began to look at. We also looked at the energetics, and that all organisms follow this pathway, that nitrate is converted to nitrite, nitrite is converted to ammonium, ammonium is converted to glutamate, and from there to other amino acids. These occur in separate compartments. Here is looking at the leaf, where the first reaction occurs in the cytoplasm. This will become important, and I'll talk about later. The second two reactions occur in the stroma of the chloroplast. The amount of energy involved is enormous, as you might expect, going from a -3 to +5. It's enormous. You have the NADP or NADPH, NADPH or NADH, the first step, and 6 reduced ferredoxins necessary in the second, and 15 reduced ferredoxin in the last. So I guess I'm getting emails. That's those are little people to keep you leaving your phones out, right? If we put it on ATP as a common currency, essentially this converting one nitrate into one amino acid takes 12 ATP. This compares with most biochemical processes that take 1 or at most 2 ATPs. It's an enormous energy cost. And so most organisms avoid it. So we wanted to try to determine how much nitrate was being assimilated in a real matter. And the technology for doing this was quite limited. So we developed about 10 independent techniques. And the one I'll spend most time with today is talking about looking at CO2 and oxygen gas fluxes from the shoot or the root, and from those gas fluxes determining how much nitrate was being assimilated. So this is the lab on a clean day. This is a chamber in which we hold a Arabidopsis plant. Here's a larger chamber, obviously, that's holding a giant redwood tree and a small giant redwood tree. This is 12 wheat plants. The bubbles are, it's just water being used as to control temperature. It's not actually, they're not swimming. So we started looking at techniques that have been done through the ages, and came upon one that was first developed by Warburg. I don't know how many of you, I guess as you get old, you get more interested in history, right? That's a sign of dotage. It's getting interested in stuff. Some of the historical leaders in photosynthesis are Warburg and Emerson. Some of you may know those names. And they started playing around, and they discovered, way back when, that closed containers, that this is known by, that we could measure CO2 fluxes versus O2 fluxes to get an idea of what nitrogen form was being used. Let's talk about respiratory quotient, because many of you, if you harken back to your studies of animals, which I don't know how long that is for some of us, that there's a thing called respiratory quotient, which is the amount of CO2 released versus O2 consumed during respiration. And it harkens back to a motto of my generation, that is, you are what you eat. And I'd like to extend that. You are what you eat. I can tell what you've eaten by the gases you're giving off. So, for example, if you had oatmeal for breakfast, a carbohydrate, then you would give off equal amounts of carbon dioxide for the amount of oxygen you consumed. Now, if you had bacon for breakfast, and we're metabolizing those fats, then you would give off less CO2 for the oxygen you consume. And this is true not only for animals like you, but for plants. So seeds that use carbohydrates for storage have a respiratory quotient as they germinate of 1, and those that store lipids as they germinate have a respiratory quotient of 0.7. So we use that as a way of determining what they're using as a nitrogen source, because when they're using nitrate as an electron acceptor rather than oxygen, they release CO2 without oxygen being consumed. That causes a shift in the respiratory quotient. And that's not true under ammonia. Taking the flip side, looking at what happens during the daylight, when in fact oxygen is being released during the light-dependent reactions of photosynthesis, and CO2 is being consumed during carbon fixation, we get a similar sort of story in the sense that CO2 and nitrate assimilation are competing for reductants, and that oxygen is being released because it's generating reductants and electrons that are being accepted by nitrate and nitrite without being used by fixing carbon dioxide. So oxygen is released without CO2 being consumed. This was done in the 30s and 40s, and what they discovered, like in chlorella, which they could do in very nice apparatus. I don't think that anyone here, except me, has ever used a Warburg apparatus. You have? Thank you, John. But when you put chlorella, which is about 20 to 25% protein, when you expose it to ammonium, you get a similatory quotient of 0.94. When you put it under nitrate, and therefore the nitrate is accepting those electrons rather than CO2, it shifts downward to 0.68, very dramatic shift. So we are using this for plants, and the reason we use it, it gives us a real-time, non-destructive measure of how much nitrate has been assimilated. Now, we started looking at both the respiratory quotient and summatory quotient in a variety of plants. This is an early paper where we looked at how much was occurring in roots with nitrate versus ammonium as a nitrate source. And what we discovered that root respiration under nitrate consumes about 1/4 of the energy in the root. We did this on shoots as well, and we found, lo and behold, that shoot respiration for nitrate assimilation, again, consumes about 25% of the energy in the plant. Now consider this, the plant is only 1 or 2% protein, but to generate that protein consumes about 1/4 of the energy of the plant. Very energy, when the nitrate source is nitrate. Now, to give you some idea of how this works and why other people haven't seen it, this is data from barley, and this is the net flux of CO2, the triangles in the blue, oxygen, which is red circles, and you can see on the left panel, which is the plants under ammonium nutrition, and it's those gas fluxes as a function of light level. Photosynthetic flux density. So photosynthetic photon flux density is what the PFD stands for. And you can see that under ammonium, the fluxes of CO2 and oxygen are about equal. Now, if you shift those plants to nitrate, you'll see that at the higher light levels, where CO2 is limiting carbon fixation, but there's ample energy for the light-dependent reactions that split off water and generate oxygen, you can see that the oxygen fluxes does exceed CO2 fluxes by quite a bit. Now, why haven't other people seen this? Nobody monitors oxygen fluxes under these conditions, under real-time conditions. So in fact, you can see that plants have an excess capacity to generate electrons and reductant using light-dependent reactions of photosynthesis, and this seems to be related to nitrate. How do we know it's related to nitrate? We looked at a mutant barley that has 2 mutations that eliminate both forms of nitrate reductase, and therefore it cannot reduce or assimilate any nitrate, and that's in this figure right here. You can see there is no difference between oxygen and CO2 fluxes if they're not able to assimilate nitrate. So this moved us into looking at carbon-nitrogen relationships in plants. And as anyone who's been awake for the last 20, 30 years has probably seen this graph as the movie Inconvenient Truth. Al Gore went up in a cherry picker to show how much this had raised. I could jump, but as a white man, I can't jump very high. But as most of you know, that CO2 levels have increased about 30% over the last century or so, and it's now pushing about 400 parts per million, or 0.04%. Now, this is quite unusual in the history of the planet, that if we go back, this is from today 0 to about 25 million years is the scope of this, and we reconstruct what atmospheres were on the surface of the Earth over that 25 million years, you can see that conditions, we haven't had CO2 levels this high for the last 24 million years. CO2 has been lower than they are today for the last 24 million years. And during that 24 million years, nothing's happened, right? Well, plants have done nothing. Now, actually, during this 24 million years came the rise of grasses. And also multiple evolution of C4 species, particularly during times when CO2 really dealt below 200 parts per million. But a lot has happened. One thing that has not happened, and that will be key for what I'll say, is Rubisco and C3 plants have not changed. That's a clue to something that is going to happen, see if it sticks in your brain. So over the last couple of decades, we've done a number of plant species and looked at how the assimilatory quotient has changed as a function of CO2 concentration. CI, the x-axis here, stands for internal CO2 concentration. And this is the change in AQ as one goes from ammonium to nitrate. And that is a measure of nitrate assimilation. So as in about monocots, dicots, angiosperms, gymnosperms, in all different types of C3 species, As CO2 concentration within the leaf increases above levels that you find today, the amount of nitrate assimilation that is there disappears. Yeah. Elevated CO2 inhibits nitrate assimilation in all these species. Going to C4 plants, the maize, monocot C4, Flavaria dicot C4, Amaranthus has a different C4 pathway than maize. You can see that CO2 levels in the leaf really doesn't affect very much the amount of nitrate that's being assimilated, but look at the scale, 0.12 versus 0.24. We'll put this all together in the last panel. Here in the lower left panel, we have that a C3, two C3, C4 intermediates, and lo and behold, the results were intermediate. And putting those all into the last panel in the lower right, you can see all the C3 plants, the mean plus the standard error. That's the purple is the C3, C4 intermediate, and C4 is the red. And you can see that there's very distinct response to rising CO2 and the different pathways of carbon fixation. Another way of looking at nitrate assimilation is shown here. We deprived plants of nitrate for a while and then gave them a pulse of nitrate. We followed how much nitrate was absorbed by the roots and then looked at how much remained unassimilated in the shoots and was able to calculate nitrate assimilation. That's the no label here for Arabidopsis, the no label here for wheat. We did this under three conditions, under ambient CO2 and ambient oxygen. That's the red bars. We did it under elevated CO2 and ambient oxygen. That's the blue bars. We did it under ambient CO2 and low oxygen. That's the red bars. You can see that as you go from ambient conditions, either changing, elevating CO2 or lowering oxygen, the amount of nitrate assimilation declines. That's also true using a different technique. This is N14 label, where we grew plants under N15 nitrogen. So we got plants that were 90% heavy nitrogen. Then we gave them a pulse of unlabeled nitrate, N14 nitrate, and looked at how much of that N14 nitrate became organic N. And that's these N14 labeled bars. Again, the same results, elevated CO2 or low oxygen inhibits nitrate assimilation in both Arabidopsis and wheat. Now, we've also done growth chamber studies, controlled environment studies on a number of species. This was sugar maple. The reason we did sugar maple is that it's been subject to a lot of experimentation, because it is a very important psychologically, if not economic, crop in the northeast of the United States, favors maple syrup. And it is one of the few hardwoods that depend very heavily on nitrate as a nitrogen source. So hopefully we'll see. We did it under ammonium as a nitrogen source, the left panels, or nitrate as a nitrogen source, the right-hand panels, ambient CO2, the top panels. The lower panels are elevated CO2 levels that we expect in the next few decades. And hopefully you can see that there's a lot of difference between the plants under nitrate and particularly the plants under elevated CO2 and nitrate. That was a poster that I have for today. And then we submitted it, and of course the one reviewer said they saw no difference between the treatments. Sometimes you just want to go out and shake them. And particularly if you look at the plants under nitrate as a nitrogen source and high CO2, plants are dying because of nitrogen starvation. And we can measure that, and they weren't open to that. Here's another species, sweet gum. We use this because it is the species used in a large-scale experiment at Oak Ridge National Labs. And we exposed it to CO2 levels that occurred 50 years ago, about 310 parts per million. We exposed them to CO2 levels that occur today, and we exposed it to levels that we anticipate in the next few decades. Again, at CO2, particularly under nitrate, as CO2 increased from levels that were 50 years ago, growth was strongly inhibited. Again, the reviewer could not see any differences. We have numbers, but numbers are just numbers. Sorry, I'm being facetious. So those are data that we gathered from controlled environmental conditions, and you might say, how does that relate to the real world? And so we collaborated with Bruce Kimball with an experiment that was done in '94 and '95 in a wheat field where they had very elaborate equipment that's called face springs, free air carbon dioxide enrichment experiments, where out of these standpipes, which you can see in more detail here and here, they monitor CO2 at wind speed and wind direction, and out of the upwind pipes, they release CO2, and they're able to control within these rings CO2 levels at about 50% above ambient levels quite accurately. When they first analyzed these plants, they found no difference between the elevated treatment and the ambient treatment. That's total N. So this is in 2 years. Okay, you can see that the red and blue lines are identical. The two treatments are identical. And we said, well, let's look, and this is partly because they were very heavily fertilized. We said, let's look at how much free nitrates in that tissue. And we found, in fact, that the plants exposed to elevated CO2, as anticipated by our hypothesis, had a much higher level of free nitrate in the tissues under the elevated conditions. So again, it affirms that CO2 does inhibit nitrate assimilation in a field growth crop, and this came out finally in Nature Climate Change this year. So CO2 enrichment inhibited nitrate assimilation in the field. Now, how important is this? Is it just ranting on? And it actually threatens the quality of food in general. And this was a study done with Sam Myers at Harvard. And we analyzed all of the, Eli out there, helped analyze all of the plant samples from all the FACE experiments that are currently going on crop plants. And the change in harvested protein means that 0 means that there's no difference in the plants grown at elevated CO2 and ambient CO2. Minus 20 means that the protein level is 20% less in the plants grown at elevated CO2. And you can see under field conditions that wheat, rice, and barley have about an 8% to 15% decline in protein when they're exposed to elevated CO2. Legumes that are using a totally different nitrogen source really are not affected very much. Potato, again, declines about 8%. And maize and sorghum less so, but highly variable, but to some degree in general. So it may have a very strong effect on the food quality that we have in the future. So how does this affect perennials? And this is all the studies I've been able to find where people have grown for longer than a few years perennial plants, and we follow the change in biomass with CO2 enrichment. Again, 0 means there's no difference between the ambient and elevated treatments. 100%, which is the full scale here, means that the biomass doubled under elevated CO2. Negative means that it actually declined under elevated CO2. And you can see that there's a stimulation of biomass under elevated CO2 in the first year or two, but that declines with time. And in my thinking, this means is that initially they're spurred on by the elevated CO2, but with time they become nitrogen deprived, and that seems to be true when people analyze the tissue. So what is the cause of this? What is the physiological mechanism involved? And this brings us into photorespiration, which has a bad reputation among scientists. So to review, for those of you who don't think about carbon fixation or photobiology very much, in simple terms, photosynthesis involves the carboxylation of a high-amplified carbon sugar, or ABP, by the enzyme ribisco to generate the 6 cycles hexose sugar. Now the flip side is the same substrate can be oxidized by the same enzyme, and when it does that, it actually releases CO2, and the energy in that 5-carbon sugar is somewhat dissipated. In fact, a lot of it is dissipated, and this is known as photorespiration. The relative rates of photosynthesis and photorespiration depend on the relative concentrations of CO2 and oxygen at the active site of the enzyme. And so that when, at current conditions, you have about 3 cycles of photosynthetic carbon fixation versus every cycle of photorespiration. Hence, when you increase CO2 concentrations above what they are today, what do you expect? Do you expect this cycle to go faster and this one to go slower? In fact, you see about a 40% increase as anticipated in the short term. It's been assumed that photorespiration does nothing but generate waste heat, and that waste heat is it's just because plants are stupid, frankly. They don't know what to do with themselves. They evolved under conditions where CO2 was high and oxygen was low in ancient atmospheres. And so this is just a problem with this enzyme that's the most prevalent protein on the planet. It can't tell the difference well enough between CO2 and oxygen, and so they waste 30 to 40% of their energy through photorespiration. And I say, this conditions that this have been occurring for the last 24 million years. Are they stupid for so long? And the answer is, I hope you can come to your own conclusions. This is the photorespiratory pathway. I've come to my conclusions, which will be obvious. This is from a review we did in our reviews about 3 to 4 years ago. And here is the oxygenation reaction of RUBP right here. There it is, circle. And as that goes up, you find there's an increase in malate production in the chloroplast, and that's transported out of the chloroplast into the cytoplasm. It's broken down to generate NADH in the cytoplasm, and that NADH generates reductant to reduce nitrates and nitrite. So we're saying this is not just coincidence, that in fact, that photorespiration is not just wasting the energy from the oxidation of this 5-carbon sugar, it's producing enough energy to produce change the OAH into malate, and that malate is just translocated out, powers nitrate assimilation, and that's why elevated CO2 inhibits this process here, because it inhibits the generation of reductant that powers it. So plants are not stupid. Nitrate assimilation depends on photorespiration. Conditions that inhibit photorespiration, such as high CO2 or low oxygen, inhibit nitrate assimilation. This is true in monocots, dicots, gymnosperm, C3 species. And why do plants do this, and why is it an advantage? It turns out, as I mentioned, most organisms do not use nitrate willingly, only under duress. So it gives higher plants a source of nitrogen that no other species really wants. It's also a source of nitrogen that's highly mobile in the soil, and therefore they have great access to it. The disadvantage is it costs more energy. But if they're CO2 limited, they have plenty of energy to devote to that purpose. Not happenstance. What this says is that my dear colleagues that have been, for the last 4 years, trying to breed for low photorespiration are on the wrong path. They don't like to do it, and they've had no success. It hasn't worked, but they still say it's going to happen. There's a review in Nature that just came out saying, we're going to do it. Of course, they have to grow the plants at 5,000 parts per million CO2, but they can do it. This is Bernie Hansen that pointed out, yeah, they can do it, but it's not. And they have to grow it at very high ammonia levels. So, yes, this is science goes on. Okay, it also says that C3 carbon fixation, that energy is not being lost, is under most conditions, most temperature is more efficient than C4 carbon fixation. And this explains why, if we were able to look out the windows here, how many C4 plants would we see? Unless we're growing corn in our little pond out there, we'd see none. 96% of plant species are C3 and continue to be C3. 95% of the biomass in the world is contained in C3 plants, not C4 plants. So the efforts to make C4 rice and C4 wheat are a fool's errand. We'd like to be reminded about that too. Another mechanism that involves, can explain some of this, is that, as I mentioned, nitrite is produced in the cytoplasm and is translocated to the chloroplast for further processing. CO2 inhibits that transport process, we've found. And as such, that process is sensitive to CO2 not only during the day, but at night as well. So this is data that we've submitted to the Royal Society and fighting them about this. This is again Rabbitopsis in wheat. absorption versus assimilation, and the Arabidopsis assimilation of wheat. As you go from, oh, that's interesting. The clear, and it's actually pink in my version. This is plants exposed to ambient CO2 day and night. This is ambient CO2 during the day and elevated at night. Assimilation strongly inhibited both species. This is ambient during the night, but elevated during the day. The last one is elevated day and night. Again, elevated CO2 at night does inhibit nitrate assimilation in both species. Strongly significant in the wheat, less so in the Arabidopsis. You can, again, some of us can see this. I don't know if you can see it. This is Arabidopsis put under nitrate nutrition where the conditions were ambient CO2 day and night, elevated day and night, elevated during the day, ambient at night, elevated at night. You can see the smallest plants. I see the smallest plants being, and the number shows, smallest plants are those exposed to elevated CO2 only at night. Again, this could be due to, seems to be because of the nitrogen, organic nitrogen content due to nitrate assimilation being inhibited by high CO2 at night. Where this comes to bear is all, with very few exceptions, all the studies of plant responses to elevated CO2 do not control for CO2 at night. All the field studies. That is, these are some of the field studies. And the reason they don't control CO2 at night is twofold. One, it costs a lot of money, and they say CO2 at night is not important. Made that assumption. And also, controlling CO2 at night is difficult because there's not a strong sink for CO2 like the plants. So it's more difficult technologically. But in fact, very few field studies are examining how plants that are exposed to CO2 elevated both day and night are responding.
Are the CO2 levels in your roots influenced?
Yeah, that's a good question. In fact, CO2 does not affect root assimilation of nitrate. And root levels of CO2 are generally much higher on the order of 1,000 parts per million. So this is a shoot phenomenon, and we're very careful when we write these things down to make that distinction.
So it's getting in through the roots at night, though? How does it get in?
What gets in?
CO2, yeah.
Gets in.
The stomates are closed?
Well, oh, yeah, stomates are closed, but as you know, closed stomates are not closed. There is enough that the internal levels of CO2 are elevated, if you were to estimate them, right? Plant responses to elevated CO2, generally in ecosystems that are dominated by ammonium, such as freshwater marsh, you can find long-term stimulation of productivity with no decline in nitrogen in those freshwater marsh species. In coniferous forests, as they mature and depend more on ammonium, CO2 that has a less of an effect, but on young forests where nitrate is more important, you find that there is an effect. Particularly in agricultural areas where nitrate is a major form of nitrogen available to plants, like wheat fields and pasture grasslands, elevated CO2 does inhibit, in the long term, inhibit productivity and the protein content of the plant food material. And if plants are in a situation where they're totally dependent on nitrate, you find a 20% decline in protein in plant material. And this has effects beyond just humans, human consumption of those materials, that pests that are dependent on those plants for their livelihood become nitrogen starved and tend to be a greater problem because they have to eat more plant material to meet their nutritional requirements. So this seemed to be like a pulling out into a lone space, but finally some people are checking this on a larger scale. And so there's been released a number of meta-analyses where they've looked at hundreds of studies. One came out in Science about 2 years ago. Another one came out in Global Change Biology a year ago, and one came out in Nature this year, where the only explanation for the results of these studies is the fact that CO2 inhibits nitrate assimilation in the shoots of C3 plants. Still, my favorite review that I got recently Despite numerous publications and one of the prestigious journals, I still don't believe it.
Somebody really wrote that?
Yep. And so it's still a battle. And what I'd like you to say that not only is it difficult to get people to change, scientists to change their beliefs in the face of information, it's difficult to get the public to change their. So if you ask the American public what they think about climate change, 70% say that it's the Earth is warming, but only 40% think it's due to human activity. Again, refusing to believe overwhelming evidence that it is human activity responsible for rising CO2 levels and climate change. So to deal with that, I'm moving into the next part of the talk, is that all the UC chancellors have signed a commitment to make climate neutrality and sustainability a part of the curriculum and other educational experience for all students. And I've developed an online course that each of you are expected to take. It's available 24/7 at the website here, and there are quizzes, and I can keep track of how you're doing. What?
Will there be questions on the talk?
Oh, yeah, there are, definitely. I'll show you some of them right in a moment. So this is available. It's been used in the last 2 years about 30,000 times. And what's more impressive that unlike most websites, people spend an average of about 6 minutes per visit, which is pretty long if you've ever looked at statistics. So not only am I getting them to go to that page, but they're spending time there. Of course, the main bulk is about half is in Davis, California, where a student is taking my class. Some of them try to watch all the videos just before the 12 hours just before the final exam. It's probably try to advise again. So I teach it here 3 times a year. UC Extension's using it for a certificate program, environmental sustainability. It's been taught several times in Israel. Today and yesterday, I was teaching it at 11 Midwest land-grant institutions as part of the Corn Cap and the Pine Cap program. So they're supposed to give their students something about sustainability, and they came to me to do it. We're negotiating with universities in Hanoi and in other parts of Vietnam and in Iran. What's very nice about online courses that I don't have to go to double spots of there to actually deliver the material. I am working on a revision of my textbook in a multimedia edition. Let's see if I can show you this quickly. So for example, here's the multimedia, and instead of having to strain your eyes to see that, when you roll your mouse over it, it pops up. And then there's a video here. Let's see if this I can play. This is a 2-minute video, so please be patient. Oh, no. I'm going to have to be quiet because these speakers are not very good.
Some of you may recognize this portrait of Napoleon. Napoleon invaded Russia in 1812. This chart, often judged the most impressive of all time, tells the story of this invasion from the French perspective. Shown is a map of Western Russia, with the path of the French advance in brown and the retreat in black. The width of the path and the numbers indicate the size of the French army at various locations. The branched points indicate where some troops separated during the advance and later rejoined the retreat. Temperatures are in red and are linked to the path of retreat. The background music is from Tchaikovsky's 1812 Overture, which also tells the story of this invasion, but from the Russian perspective. The Grande Armée of France crossed the Niemen River from Poland into Russia on June 23, 1812, with nearly 450,000 troops. The Russian army avoided major engagement with the French and retreated further and further into Russia, scorching the earth behind them. Thus, the French found it difficult to find food for themselves and their horses. The Russians finally engaged the French in battle outside Moscow on September 7, 1812, in the Battle of Borodino that resulted in approximately 44,000 Russian and 35,000 French casualties, perhaps the bloodiest day of battle to that time. After this battle, the Russian army withdrew further east of Moscow. Napoleon entered the city triumphant. He assumed that the fall of the Russian capital would end the war, but the Russians burned the city rather than capitulate. After a month in Moscow, Napoleon became concerned about loss of control back in France and decided to return with his army to his homeland. Here comes the interesting part, the part about climate. Russia has a cold climate. This should surprise no one. By the end of October, when the French troops were retreating back to their homeland, temperatures were dropping below freezing. By the beginning of December, when these troops again approached the border between Russia and Poland, temperatures were below minus 35 degrees Celsius or minus 31 degrees Fahrenheit. The 450,000 brave souls that began this campaign had dwindled to 10,000. Only 1 in 45 survived.
Okay. So the idea is to, I have videos with most of the figures explaining what's going on. You can, this is minimum sea ice, it's not very well displayed here in the projector, but this is in 1979, and this is 2 years ago, and you can go back and forth and you can see how much it's changed just by flipping your mouse back and forth. There's data visualizations, again. The idea is how do we make the textbook be dynamic enough to hold the interest of a modern audience, engage the people actively. Thank you. Yes.
So the slide that you had about the perennial slide, decreasing their carbon fixation over time.
Yes.
Do all of those trees fix their nitrate in their shoots?
It's a very good question, one that drove me into this line of research. To what extent do plants of any life form depend on root versus shoot assimilation of nitrate? And the answer is it's quite variable among species and within species with conditions, and the answer is not clear. One thing that will shift it is we found that elevated CO2 actually stimulates nitrate assimilation to the root, as you might expect, because it's not sensitive to CO2. And that's one way plants can compensate for rising CO2, inhibiting it in the shoot, is that they do more in the root.
But it's cheaper in the shoot.
It's cheaper in the shoot because they're using energy generated from photorespiration for that purpose. Root, they have to actually transport additional carbohydrate for that purpose. 12 ATPs per nitrate assimilated into amino acids. Good question, thanks. Yes?
You showed a graph of decrease in protein harvest under higher CO2 levels.
Mm-hmm.
Can you back project and make claims about protein levels 1,000 years ago at lower CO2?
As anyone who does work in this, as we do, you know there's a problem that the genotypes that we have been growing have changed, and their protein qualities have also changed. So we can do it in chambers where we take the same genotype and expose it to various CO2 levels and see how that affects protein levels. But to look at historical data, people who've done that, it's complicated by that change in the fact that people aren't growing the same genotypes they did at one time. For good reason. That's why you have a job. That's why the Hudson.
Good. Anyone else?
Forest ecosystems are NH4. Is that because you get more microorganisms involved in the ecosystem?
Particularly in some forest ecosystems like coniferous forests, they tend to have very acidic soils. And it turns out that the process of converting ammonium into nitrate, called nitrification, which you probably know, is sensitive to environmental stress. And particularly acidity tends to inhibit that process. There's only 2 genera of bacteria that actually do nitrification. And so the ammonium that's generated in matter is not nitrified, so therefore it's more prevalent than in those cases.
It could be also the ectomycorrhizal component is very high in forest ecosystems compared to sagebrush, where they don't
Good point.
exist. Yeah, and they do carbon cycling.
Yeah, most fungi do use ammonium to a large extent and avoid nitrate for the reasons of the energetics. And so when they transfer, this is still an open question that maybe Allison can help me with, it's thought they do transfer either ammonium or amino acids to their hosts.
You mean ectos, mycorrhizae?
Yeah,
The amino acids, yeah, to some extent.
it's still an open question,
But definitely, yeah, ammonium, not nitrate.
not nitrogen. Great, thank you
Good,
all.
well, thank you, Arnold, very much.